Hello, everyone. My name is Jörn Schattenberg. I'm the director of the Metabolic Liver Research Program, and it's my honor to be presenting during this symposium of the Swiss Nash Association for June Nash Day, International Nash Day. Unfortunately, I cannot join you in person today. However, I do have my Nash Day cup of coffee here, and I'm sitting at home recording this lecture on FGF-21 analogs, which I hope you'll enjoy. And please feel free to reach out to me if you have any questions that arise in the context of my talk. So these are my disclosures. And let me start by briefly touching on the prevalence of the disease. Now, you heard by the different speakers that this is a relevant disease. My take at that is that I'd like to highlight that it's a slowly progressive disease. While there is a clear link of NASH and inflammation to fibrogenesis and cirrhosis, this is nothing that happens overnight in most cases. So we're looking at a heavily comorbid population with metabolic cofactors, and these patients progress over years to end-stage liver disease. Now that's relevant, and we've done one analysis in a German general population, and if you explore the fatty liver index as a surrogate for advanced fibrosis and take the cutoff of 2.67, you see that 1.1% of the German population are above that threshold in the context of NAFLD-associated disease, and that's a high number of patients affected. We know from the Nash CRN, and that paper has been published last year in the New England Journal, that it's particularly the advanced disease stage population that achieves endpoints F3 pre-cirrhosis and F4 cirrhosis. And of course, non-alcoholic steatohepatitis is not a standalone liver disease. It's a disease that develops in a severely obese, severely metabolically diseased individual. They develop cardiovascular disease, they develop dementia, colon cancer among cancer etiologies that are increased. There's chronic kidney disease. And interestingly enough, the inflammation that is centered in the liver or originates in the liver affects these comorbid conditions. Most of us in clinical trials is shown as association, but there are also mechanistically links that have been demonstrated in animal models. So the question is, can we focus on inflammation as a treatment target for NASH, being responsible for extrahepatic and hepatic phenotype? And how can endocrine signaling pathways, we're talking about FGF-21 today, exert a potential benefit? So let me detail the therapeutic strategies as of today, June 2022, briefly. I think we all agree that there's a baseline concept that patients with metabolic disease have to modify their metabolic risk profile, reduce lipotoxicity, achieving weight loss, and as such, improve glycemic control. The question is, if we're using pharmacotherapy, can all of this be achieved by a single agent? And this is a review that has come into the ages, but it nicely shows that we're thinking in different categories or pillars here. There's anti-metabolic drugs, there's anti-inflammatory drugs, and anti-fibrotic drugs. The question is how to best combine them to achieve maximum benefit, and potentially this is different in different types of patients. Now, for the purpose of the talk today, I'm going to be focusing on the FGF-21 analogs that are deeply and intimately involved with the lipid tissue in regulating inflammation, or more broadly, metabolic inflammation. You can see here a pipeline of MOAs and compounds that are being explored in phase 2 and phase 3 trials. And I've highlighted the endocrine FGF class to give you an idea of their positioning in the overall landscape. Now, let's dig into the class of FGF-21s a little bit more deeply. FGF-21 is an endogenous metabolic hormone that responds to stress. Importantly, it regulates energy expenditure, glucose, and lipid metabolism. And as such, the white and brown adipose tissue and the liver are crucial organs, effector organs. Now, interestingly enough, it also regulates satiety and food intake. It affects the vasculature and the musculature. It has been shown to reduce liver fat and then also has impacts on liver fibrosis. Now, the downside of using FGF-21 as it is its short half-life, which is below 2 hours. So whenever you want to use this as a pharmacotherapy, there have to be modification introduced to ensure a more lasting effect. Something that comes from the preclinical field, I'm just showing you this one study here that's what's done in mice, is interestingly enough, if you have mice and put them on a low protein diet, restricting protein, you can see here in picture B that the red line has an increased overall survival longevity, meaning that certain dietary modifications will help to improve your lifespan. Now, if you knock out FGF-21, and that has been done in this graph C, that loss, that benefit in overall lifetime from protein restriction is actually lost again, telling us that FGF-21 sits very central at the control of metabolic parameters and even survival under the circumstances of dietary modifications in these mice. Now, I've been asked to focus on FGF-21. There's an interesting FGF-19, and they're similar but not the same. I'm not going to be discussing aldafermin today. There is a number of FGF-21s in development, but for the purpose of this talk, I'm going to be focusing on the Bio-89 compound 100, or pegozafermin, and efruxifermin. Let's start with a pegozafermin. Now, the data I'm going to be showing you hasn't been published in full paper, but is taken from conference presentations. Here, FGF-21 has been modified by adding a glycopegylated compound to increase its half-life, and by thus increasing the half-life of up to 55 to over 100 hours by a single dose. Now, importantly, low nanomolar potency is maintained for the FGF receptors 1C, 2C, and 3C, but not for the FGF4 receptor, which is that receptor signaling pathway that actually is responsible for increasing LDL levels in response to FGF-21. Signal transduction, you can see that here, is achieved through binding of the FGF receptor 1 in most cases, and then co-activation with beta-klotho. Now, the first study, or the phase 1b/2a Nash trial design, where I'm going to be showing you some data, is a multiple dose study here. You can see that patients with histologically confirmed Nash or phenotypic Nash based on imaging biomarkers have been included. There was predominantly safety readouts, but also secondary endpoints, relative changes in liver fat, and metabolic parameters assessed. And you see here that 13 weeks of treatment versus placebo achieves an overall relevant reduction in intrahepatic liver fat assessed by MRI-PDFF. Even if you give it Q2 weekly, there's a robust reduction in intrahepatic liver fat. Now, the trial has included patients that are of particular concern to me, and every once in a while in clinics I do have these patients that have particularly high triglyceride levels, defined as above 500 mg per deciliter. And here you can see this is the subgroup analysis of the patients, which also achieved a reduction in liver fat with the high triglycerides. And the triglycerides normalization rate is actually significantly higher, highlighting that the pegozafermin is able to even achieve that reduction of liver fat and triglycerides in a difficult to treat patient population with high triglycerides at baseline. Here are some metabolic parameters that have been assessed in that study. You can see that insulin sensitivity measured by HOMA-IR, glucose levels or A1C increases in this short time frame over 13 weeks. There's even a slight weight reduction and beneficial effects which are explained through the functioning on the adipose tissue, or improved function of adipose tissue with FGF-21s is an increase in adiponectin. What about safety? One of the effects that I mentioned is the central nervous regulation of appetite and satiety. So you can see that actually patients in this trial, looking at the pooled Bio-89 100 data over all arms, the participants reported increased appetite, while the GI side effects were comparable to placebo in this trial. No severe adverse events were noted. So let's shift gears and talk about the second FGF-21 compound, efruxifermin. Now here, the setup is a little different. You can see the molecule has been designed to include two molecules which are combined through a linker, and they are able to bind to FGF receptors and beta-klotho, and the affinity to bind to beta-klotho is actually improved. So key attributes of efruxifermin, like the Bio-89-100 compound that I just detailed, it mimics FGF-21 biological activity. The half-life is likewise significantly improved, and there's a balanced potency again against FGF receptor 1C, 2C, and 3C, but not the 4, which is leading to LDL increases. And here's the study design, and that study has been published in Nature Medicine by Stephen Harrison as the first author. You can see it's a 4-arm study comparing 3 different doses of efruxifermin with increasing dosage, 20 patients per arm over 12 weeks. Importantly, the study design is set up that at 12 weeks, there's an MRI-PDFF done, and in patients that do achieve a pre-specified reduction of intrahepatic liver fat, they are actually eligible for post-treatment biopsy, which has been done in a subset of patients, and I'm going to show you that data in terms of responder analysis. So here are the key data from that phase 2 trial, looking at the efficacy, the primary endpoint, MRI-PDFF measured intrahepatic fat reduction. And you can see that there is a high number of patients achieving a more than 50% reduction of MRI-PDFF relative reduction, or more than even 70% of MRI-PDFF reduction in the with a dose-dependent effect, and that was not seen in placebo. Normalization of liver fat content, which is the maximum that can be achieved with MRI-PDFF readouts. In the high-dose group, 67% of cases versus only 5% of the cases on placebo. And this is the responder analysis at week 16. I'm showing you histological data on Nash resolution without worsening of fibrosis. Importantly, only in those patients that had a positive response to MRI-PDFF at week 12. So only if you had a more than 30% decrease in MRI-PDFF at week 12, you were eligible for biopsy. And you can see that this happened in 1 out of 2 placebo patients and in 19 out of 40 efruxifermin patients. And looking at the different treatment arms, this was fairly evenly distributed. Now, looking at fibrosis improvement after 16 weeks, again in the responder population, detailed or defined as a fibrosis improvement of more than 1 stage without worsening of Nash. None of the 2 placebo patients that did respond, qualify for MRI-PDFF response, did achieve a fibrosis improvement, while this was seen in 19 out of 40 efruxifermin patients. And here, no clear dose signal was evident from this small study. When only looking at the F2, F3 patients, this is detailed here, and even a significant number of patients achieved a 2-stage improvement in fibrosis on the responder analysis, showing that there seems to be a potency if you're responsive to drug and reduce MRI-PDFF or intrahepatic lipids, then you're even capable of improving 2 stages of fibrosis, and that was happening in 11 of those patients. How about safety and drug treatment-related emergent adverse events? You can see, as expected, there's a certain GI side profile, diarrhea, nausea, and increased appetite, all numerically higher compared to placebo, but also abdominal pain and changes in bowel movement. There was no death, but there were 6 patients on drug efruxifermin that actually had to discontinue related to treatment emergent adverse events, with 2 serious adverse events in the treatment groups, as detailed on this slide. Now, let me show you some additional metabolic parameters here. You can see that patients at week 16 did show improvement in H1C. And if you look at all patients, there's dose-dependent effect with a 0.5 point reduction in the 70 milligram groups. And if you only look in type 2 diabetes patients, that effect was even more pronounced with a decrease of 0.9. C-peptide levels have improved, and that was comparable in all patients and the subgroup of patients with type 2 diabetes. And here you do see the changes in lipid levels with benefits on triglycerides, increased HDL, and unchanged LDL cholesterol levels. Now that's important because, as I said before, activation of FGF receptor 4 will give you increasing LDL levels. Now, with that, I'd like to come to a conclusion and say and revisit actually the treatment paradigms we're discussing for NASH today. As I said in the beginning, I think there always have to be baseline modifications in patients. The question I raised was, is one drug good enough to then do the job we want to see? Is one single therapy enough? I think the FGF-21s, with the data that I just showed you, have some promise in managing many targets in this pyramid, improving metabolic health, potentially not worsening body weight, improving liver outcomes, and an acceptable side AA profile. So it will have to be determined if such a drug that is an injectable at this time can be used potentially in combination with patients that are more advanced and need urgent treatment. Open questions and conclusions from my side. I think, as I mentioned in the beginning, chronic fibrosing inflammatory liver disease are slowly progressive. However, there's a relevant subgroup of patients that need urgent treatment to avoid that expected increase in end-stage liver disease. The endocrine modulation has a good rationale. FGF-21 could be one therapeutic target to be achieved that. And I think we'll identify FGF-21 as a potential combination partner in the future. Now with that, I'd like to thank you for your attention. Thank you for following us during International NASH Day. If you have any questions, please reach out, or you can reach me by email or on LinkedIn and Twitter, and I'll be looking forward to follow up with you. Thank you so much and have a good day.